Dear students, Namaskar. In this lecture, I am going to teach you the chapter number 2 of class 11 biology, that is biological classification. So I am Dr. Nutan Prakash Vishwakarma, having more than 14 plus years of teaching experience. I taught thousands of students like you, and I hope my lecture is going to definitely contribute in the knowledge of the biology and to fulfill your aspiration to become a doctor. So in my previous lecture, I discussed about the chapter number 1, the living word. In that, I told you there is different concept of the classification. There is the different categories, the kingdom, the phylum, the other things, class, order. Okay, so let's understand that categories in detail. So biological classification is not the new process. Since the human civilization language was developed, humans started doing the classification on the basis of their use, that how many plants are there in their surrounding which they can consume there as a food. They can use the cloth over from it. How many plants are there? What are the different plants they cannot use? What are the different animals they can harm to human? And how many animals are there? The list of the animals which may be the beneficial for the human use. So that is the very preliminary type of the classification of the organism. So first bit scientific classification was done by one scientist, Aristotle, philosopher, scientist, and he used some morphological characters which still we use. He divided plants in three parts: the tree, shrub, and the herb. The plant whose height is more, we can call it as a tree. If it is the middle level, you can call it as a shrub. And if the stem is very weak, there is no concept of the wood here. You can see here the example of the mint. You can grow in your pot. So stem is very weak and size is very less. So that you can keep under the herb. And that this classification still we use in our daily life. He also does the classification of animals, and he divided animals in two groups. The one who have the red blood and another who don't have. So there is two category. We see red blooded animal and colorless blood animals. Say for example, we have the blood of red color, but if you kill a ant, the blood is colorless. Grasshopper, it is colorless. All right. So later on, one scientist, Carolus Linnaeus, he did more scientific classification. And what he did, he divided all the organism into two kingdoms. So I told you in the first chapter that kingdom is the topmost rank. So he divided all the organism present in two kingdoms. One kingdom is known as Plantae, another is Animalia. There is two animals. But the problem with the two kingdom classification, that prokaryotic bacteria, obviously that is prokaryote, and cyanobacteria, both are prokaryotes. Either it is prokaryote, either it is bacteria or cyanobacteria. And plants are what? Eukaryotes. So these prokaryotes, bacteria and cyanobacteria and plants, you have to keep in single kingdom Plantae. So problem is both prokaryotes are also there and eukaryote is also there. So this is problematic. We cannot keep. If you talk about the heterotroph and autotroph, heterotroph means very simple. The organism which are dependent for their food, they are dependent on other animal or on other plants. Just like take our examples. We are taking food from the plants or from the animals. Autotroph means the organism which can produce their food by themselves. Like what? Like plant do the photosynthesis. So they can prepare their own food. They are not dependent on any other plant to eat or any other animals. So all the photosynthetic organisms are autotroph. All the animals, they are dependents on the other animal or plant. So here you can see the heterotroph and autotroph, fungi and plant. So plant is autotroph, right? And fungi is heterotroph. So here there is no place of the fungi. So fungi and plant being kept in the single kingdom, that is known as planting. Again, it is problematic. You cannot keep. If you talk about the unicellular, whether the organism is unicellular or multicellular, take the example of the protozoa. So protozoa is unicellular. However, we people are the multicellular. So both are kept in the kingdom Animalia. You cannot keep. Unicellular is also in kingdom Animalia. We people are also in the kingdom Animalia. So again, there is the problem. This is the protozoa. Another problem with this two kingdom classification, some of the lower organism, example you can see the Euglena. Euglena shows both the characters, plant as well as Animalia. So it has both the characters of the plants and as well as the Animalia, animals. That's why it is known as connecting link between plant and animal. We will study the Euglena in detail once I will discuss about the Protista. Don't worry. As of now, I think means Euglena did not get a proper group. So there was need for another classification, another kingdom. So now three kingdom classification came into existence, and that was proposed by Ernst Haeckel. So now there is three kingdom: kingdom Protista, Plantae, Animalia. Good one. So Plantae, Animalia. But here again the problem is there is no separation of prokaryote and eukaryote. Again, because all the Protista, they are unicellular eukaryote, and all the bacteria, they are prokaryotes. Still all the prokaryotes and the bacteria kept in the kingdom Protista. So again, this is not sufficient. Both unicellular and multicellular organisms are classified under Protista. So again, there is the problem. So there is need of separate kingdom. So one more kingdom was proposed by that was four kingdom classification, Herbert F. Copeland. Now it is bit better. How it is better? All the bacteria, prokaryotes, you can keep in the Monera. Plantae is clear. Animalia is clear. All the unicellular eukaryote, although they are eukaryote, but they are unicellular, you can keep in the Protista. Good one. So now it is bit better than the three kingdom classification. But still there is the problem, limitation. What is the limitation? Fungi is still included in the kingdom Plantae. And in the Plantae, you know, kingdom Plantae, all the members are autotroph, but fungi is heterotroph. So autotroph and heterotroph you have to keep in single kingdom, which is not acceptable. So there is need of different kingdom classification. So five kingdom classification was proposed by R.H. Whitaker. You have to keep remember. So what he did actually, he divided all the organism into five kingdom. All the bacteria means prokaryotes were kept in kingdom Monera. All the unicellular eukaryote being kept in the Protista. Animalia, all the animal, all the plant kept in the Plantae, and the fungi were kept in the fungi. Now it's better. So while he was making the five kingdom, he considered some of the five characters. What character he considered? First, complexity of cell structure, whether the cell is prokaryote or eukaryote. Complexity of body structure, whether it is unicellular or multicellular. Mode of nutrition, whether organism is autotrophic or heterotrophic. What are the types of reproduction, whether sexual or asexual or other modes, and what the evolutionary relationship means, phylogenetic relationship among organisms. Means over the period of time, what is the relationship, because evolution is the continuous process. So what is the evolutionary relationship among organisms? that also he considered. So you can check it out here. 5 kingdom classification. First, you have to ask the question whether the cell is prokaryote or eukaryote. If it is prokaryote, kept all those prokaryotes under one kingdom, Monera. If it is eukaryote, again two questions, whether it is unicellular or multicellular. So if it is unicellular, you have to keep in the kingdom Protista. Done? Miss R.H. Whittaker have done it. If it is multicellular, again two questions, if cell wall is absent or present. If it is absent, kept in the Animalia. If cell wall is present, now you have to see the mode of nutrition. If it is autotrophic, they comes under the Plantae, and if it is heterotrophic, you can keep in the Fungi. So now this is a good one. But there is certain limitation, still there is certain limitation. So in the Monera, these are the things that we are going to discuss in detail. Like Monera have all the prokaryotes, like Eubacteria will discuss, Rickettsia, Actinomycetes, blue-green algae, Archaebacteria, Mycoplasma, that all I will teach you while I will discuss about the kingdom Monera. Then in the Protista, all the unicellular eukaryotes are there. I will discuss about the dinoflagellate, the diatom, the euglenoid, the slime mold, and protozoa. All the fungi we'll discuss under the kingdom Mycota, the true fungi, and the Plantae. We have to discuss algae, bryophyta, pteridophyta, gymnosperm, and angiosperm. Here it is written as BGA. The full form of BGA is blue-green algae. Don't get confused. Name is written blue-green algae, but actually it is not algae. I will discuss in the kingdom Monera. In the Animalia, all the multicellular animal, as already told you, there is still certain limitation of the 5 kingdom classification. 5 kingdom classification, the virus that you know, they have the mixed character. They are showing the living character also and non-living character also. That's why virus is considered as connecting link between, sorry for my sore throat, connecting link between living and non-living. Viroids and lichens. Lichens are what? The association of fungi and algae is known as lichen. We'll discuss lichen later on. So virus and lichen still don't have any place in 5 kingdom classification. Now the next one, most advanced one, that is known as 6 kingdom classification. That was proposed by Carl Woese. Now we have 6 kingdom classification also. The scientist Carl Woese, he introduced 6 kingdom classification in 1977. He introduced a very new concept. All the organism, he put it into 3 domains. The first domain was known as archaea. The second is the bacteria, and the third is the eukaryota. What it did actually, all the prokaryotes, which previously as per the 5 kingdom classification were kept in the Monera, he divided the Monera into 2 kingdom, 2 domain, archaea and bacteria. And the basis of the separation of the Monera into archaea and bacteria was the 16S rRNA gene present in the prokaryotes. Although the archaea and bacteria, both domains of organism are prokaryotes, but there is a difference in the 16S rRNA gene. And archaea are known as the oldest living beings. We'll discuss about the archaea in a couple of minutes. So you can see there is 3 domains, the archaea, the bacteria, and the eukaryota. Previously only Monera, all the organisms as per the 5 kingdom classification, maybe it is archaea or bacteria were kept in the Monera, but now there is 3 domain, archaea, bacteria, and under the domain there is 1 kingdom that is known as Archaebacteria, and in the domain bacteria the kingdom is Eubacteria, and all the eukaryotes like Protista, fungi, Plantae, and Animalia comes under the domain Eukaryota. Alright, so this is enough to understand the six kingdom classification, but as per the NCERT, we have to study about the five kingdom classification in very detail. So as you know, there is five kingdom: Monera, Protista, Fungi, Plantae and Animalia. So I am going to discuss the same, all the kingdoms in that pattern only. So first we'll discuss the kingdom Monera. I already told you that all the prokaryotic organisms were grouped together under kingdom Monera. And bacteria are the sole member of kingdom Monera, means in the Monera you will find only the bacteria. Maybe it is Archaebacteria or it is the Eubacteria. Cyanobacteria as well. So Antony von Leeuwenhoek is known as father of bacteriology. Robert Koch is known as father of modern microbiology. And most bacteria are most abundant microorganism. They find everywhere. Anywhere you may find the bacteria. Even bacteria you can find in the extreme habitat where normal bacteria cannot survive. Some special type of bacteria, and that special type of bacteria is known as Archaebacteria. For six kingdom classification, I kept in different one, that was known as Archaea. But as per the five kingdom classification, the Archaebacteria are also the part of the kingdom Monera. So Archaebacteria always lives in the extreme habitat, means where hot spring is there, desert, snow, deep ocean, where very few other life forms can survive. So there is special kind of the bacteria. We'll discuss in couple of minutes what are the specialty of the Archaebacteria. So many of them, all the bacteria, live in our other organism as parasite, and some are free living also. As per shape of the bacteria is concerned, various form of shape is there. Like some bacteria are spherical in nature, they are known as cocci. Some are rod shape, we call them bacilli. Some are comma shape, we call them vibrium or vibrio. Some are spiral shape, comes under the spirilla. So different type of shape we found for the bacteria. As per the nutrition is concerned, bacteria have the most diverse nutrition. Some of them are autotrophic, means they can synthesize their own food, and majority are the heterotroph, means they are dependent over other animals or plants for their nutrition. So just a minute. Yeah. So autotrophic again of two types. Number one, either they can photosynthesis, can perform the photosynthesis, or they are dependent on the chemicals for their nutrition. And vast majority are autotroph, heterotroph. Either they are saprotroph, means they can get the nutrition from the dead and decaying, maybe it is animals or plants, or maybe parasite, they live on the other organisms. Now if you classify the Monera, kingdom Monera is divided into three subkingdom as per the five kingdom classification: the subkingdom Archaebacteria, the subkingdom Eubacteria, and the subkingdom Cyanobacteria. So now let's study about the true bacteria, Eubacteria. Now let's discuss the special type of the bacteria, that is known as Archaebacteria. Archaebacteria always being found in the extreme habitat. That's why they are known as extremophile. Now let's understand what is extreme condition. Extreme condition means the hot spring. Hot spring, I think you might be knowing about it. Once you go to visit to the Uttarakhand, Yamunotri or Gangotri, there naturally hot water coming outside of the earth. So you can find here the hot spring. The boiling water is there, and surprisingly, scientists, if you take the sample from here, there is the bacteria, which is not normal. They live in the hot spring, the hot condition. That's why they are known as thermophile. They are temperature loving. Even if you take the sample from the ocean, from the sea water, I mean marine water, there also bacteria are present, because the normal bacteria cannot survive in the salty condition, but these bacteria survive. That's why they are known as extremophile. What is the reason? How normal bacteria cannot survive in the extreme condition, but Archaebacteria can survive? The difference is the cell wall, the structure of the cell. The cell wall of bacteria is usually made up of peptidoglycan, but the cell wall of archaebacteria is made up of pseudomurine, which make it suitable to live in the extreme condition. And archaebacteria, they are also autotrophic in nature, means they can synthesize their own food. Some of the example of the extremophile, the thermophile, means the archaebacteria, which are thermo means temperature, phile means loving, means they can survive between 41 to 122 degrees centigrade. Even in the boiling water they can survive. The name example is Thermus aquaticus. Thermoacidophiles means temperature, thermo means temperature, acido means acidic conditions, philes means loving, means those archaebacteria who can survive in the heat and acidic condition, they are known as thermoacidophiles. The genus Thermococcus is the example. Methanogens means what? The methane, the bacteria which produce methane as a byproduct. What methanogens being found? Because normal bacteria cannot survive in the methanic condition, but all the ruminants, means the animals who can, jugali, you might be knowing it. Whenever you see the cow in the resting stage, she started mastication, just like we are chewing the chewing gum, right? Likewise it's doing. So they're known as ruminants. There is the bacteria present in their in the gut, and that bacteria produce the methane gas, and that methane gas ultimately that bacteria are present in the dung, maybe it is cow or buffalo, and if we make the gobar gas, that is known as gobar gas, means one structure is there where the cow dung being thrown and the methane can be taken out, and that methane we can use for the cooking gas, right? So those bacteria which produce the methane as a byproduct, they are known as methanogens. Many genus is there: Methanobacterium, Methanobacillus. Some bacteria can survive in the high salt concentration. Example is Halobacterium, Halococcus. They also have one reddish pigment that is known as bacteriorhodopsin. With the help of that, they can tap sunlight and form the ATP directly. Barophiles, the bacteria who can survive the high pressure environment to grow. Example, Halomonas salaria. Psychrophile means the temperature, the bacteria which survive in the extreme cold condition, means minus 20 degrees centigrade to plus 10 degrees centigrade, such as this condition found in the polar region and the deep sea. So if you sample from there, you will find the psychrophiles. So these are a special kind of the bacteria which can and be kept in the subkingdom Archaebacteria. They are a special kind of the bacteria. The another one, the subkingdom Eubacteria, which is normally known as bacteria, true bacteria. The bacteria are surrounded by two protective covering. The outer is the cell wall. You can check it out here. There is the cell wall of the bacteria, and after that there is the plasma membrane of the bacteria. The cell wall, as I already told you, it is made up of peptidoglycan in the case of the normal bacteria. However, in the case of the archaebacteria, it is made up of pseudomurine. Alright. Few bacteria, the exceptions are always there. The few bacteria which don't have the cell wall. We'll discuss later on about this bacteria also, that is mycoplasma, that don't have the cell wall. On the basis of the thickness of the cell wall of the bacteria can be divided into two parts. That was divided by Christian Gram. So he divided all the bacteria present in the nature, either they are gram positive or the gram negative. What is the difference in gram positive and gram negative bacteria? If the cell wall, means the peptidoglycan layer of the bacteria, is thick, we kept in the gram positive bacteria category, and if the peptidoglycan layer is thin, then it is known as gram negative bacteria. Alright, the size of the bacteria is 3 to 5 micrometer. Heterotrophic bacteria are most abundant in nature, and majority are important decomposer. There is many ways of the reproduction in the bacteria. Bacteria may reproduce mainly by fission, means one bacteria can divide into two bacteria. And they also have a sort of sexual reproduction, although it is not true. There is no concept of the male bacteria and female bacteria. But since one bacteria transfers its DNA into another bacteria, that's why we can call it sort of sexual reproduction, although it is not true reproduction. So reproduction is of two types. Asexual means binary fission. Under favorable condition, once plenty of food is available, all the conditions are optimum. In that case, bacteria can divide into two. The sexual reproduction is of again three types. I already told you this is not the true kind of the reproduction, but you can say it is the sexual reproduction. There is three ways: the conjugation, the transformation, and the transduction. Sometimes under unfavorable condition, bacteria also may produce the spore, the special kind of the structure which when favorable condition come will develop into new bacteria. Nutrition I already told you, maybe are two types, autotrophic and heterotrophic. Photoautotroph is of two types, photoautotroph, chemoautotroph, means bacteria can do synthesis, photosynthesis, but interestingly, in the bacterial photosynthesis, as in the plant photosynthesis, oxygen comes, oxygen liberate. Here in this case, bacteria does not liberate the oxygen. So you can call it as a anoxygenic photosynthesis. Example you can find the chlorobium, the rhodospirillum. Chemoautotroph means they use chemical energy like nitrobacter and nitrosomonas. Bacteria are heterotroph, two type, parasitic means they cause in plant and animals disease. Example is Vibrio cholerae. The saprotroph means majority of bacteria are decomposer. Example is Pseudomonas. Some bacteria also reside in the symbiotic condition. If you get the plant of Cajanus cajan or arhar, you can say, or pulses, any pulse. You may take urad, you can take moong or gram. So once you pluck the plant from the soil, you will find there are the nodules in the roots. In those nodules, bacteria are present. Thus, bacteria is known as rhizobium. They live in the symbiotic form with the plant. Both are being benefited. The benefit of the bacteria is bacteria get the shelter in the plant. And what plant get in back? Nitrogenous, atmospheric nitrogen is converted into nitrate, and that nitrate bacteria give to plant for its growth. So plant give the shelter to the bacteria and bacteria gives the nitrate to plant. So both are mutually benefit. So that is condition is known as symbiosis. As per economic importance is concerned, there is lot many applications. Bacteria convert milk into curd. The name of bacteria is known as lactobacillus. We can produce the cheese with the help of the bacteria. Example is Propionibacteria cerevisiae. Alcohol production with the bacteria Gymnosus mobilis. Many bacteria are able to fix atmospheric nitrogen, just like I told you, that is rhizobium. Escherichia coli also help in the synthesis of vitamin B and K. With the bacteria, you also may find various antibiotics, because just like we have the immune system, no? So what our immune system does, if any foreign bacteria or virus or anything, it try to come in our body to infect, our immune system fight with it. But since bacteria is of single cell, so it cannot have an immune system. So they can produce some compound, and that compound kills other species of the bacteria, because there is always no competition for the food. So if we pure those kind of the compound, we can use them as a medicine. So like a streptomycin that was produced by Streptomyces griseus, and these are available in the form of the injection and tablets. Chloromycetin, it is produced by Streptomyces venezuelae. Terramycin, Streptomyces ramosus. Tetracycline, the well-known, the Streptomyces aurifaciens. So from bacteria itself, we are getting many antibiotics. Plant disease, the bacteria produce many diseases also, like citrus canker. You can see in the citrus fruit, like orange or the lemon, you can find the canker. Here you can see the rice, the leaf of the rice is, it's known as blight of rice. It's the rice leaf is blighted, right? So this is known as blight of rice. It is caused by Xanthomonas oryzae. Here you can find the crown gall disease, that is caused by Agrobacterium tumefaciens. Here you can see the tumor kind of the structure is formed. As per animal disease is concerned, there is lot many bacteria which is causing the disease in us, like pneumonia, that is caused by Diplococcus pneumoniae, means lungs get infected in this case. The typhoid fever, that is caused by Salmonella typhi. Cholera, Vibrio cholerae, in this the diarrhea and vomiting takes place with the infection of the Vibrio cholerae. The tetanus, that is caused by Clostridium tetani. The gonorrhea and syphilis, they are both sexually transmitted diseases that are caused by Neisseria gonorrhoeae and Treponema pallidum respectively. The diphtheria, it is also known as Kali Khasi in Hindi, that is caused by Corynebacterium diphtheriae. The leprosy, that is caused by the Mycobacterium leprae. Now the third subkingdom, that is known as cyanobacteria. Cyanobacteria, we also call them as a blue-green algae. Sometimes the short form you will find as the BGA, and it is also known as Cyanophyceae or Myxophyceae. The cyanobacteria are unicellular. It may be colonial sometime, means they live in the colony, or they are in the filamentous shape. Shape is just like a filament. Cyanobacteria you may find in the freshwater or marine or terrestrial algae anywhere. They are photosynthetic oxygenic bacteria, means they have the chlorophyll A and they can produce the oxygen during the photosynthesis. However, I was talking about the eubacteria. I told you that they cannot produce the oxygen. All right. And cyanobacteria were the first organism that produced oxygen on the earth. Don't get confused with the blue-green algae. It is not true algae. It is actually bacteria. It seems like the algae. That's why we can call them roughly as a blue-green algae, but it is not algae. It is cyanobacteria. Different forms of cyanobacteria I told you. It may be unicellular like spirulina. You can see the spirulina powder is now these days in the market. They are edible because they have the large amount of proteins. So all the vegetarian people can have the spirulina powder. Colonial bacteria live, cyanobacteria live in the colony, like Anabaena. You can see here, they live in the colony. Although they are unicellular, the Microcystis, they also live in the colony. Some bacteria are filamentous in shape. You can see here the filamentous, like 1 bacteria, 2 bacteria, 3 bacteria. They are arranged, all are unicellular, but they are make the kind of the filamentous. Example is Oscillatoria, and the filament of blue-green algae is known as trichome. This filament is known as trichome. All right. Some of these organisms can fix atmospheric nitrogen in a specialized cell, just like this is the Anabaena, and in Anabaena some cells is specialized. In the nostoc, some cells are specialized and they start fixing the atmospheric nitrogen into nitrate. Many bacteria also form in the mainly bacteria, the cyanobacteria may found in the free living as well in the symbiotic form. If you see in the free form, they may found in the water or in the soil, just like Anabaena, nostoc and Alocera. All these 3 cyanobacteria you may find in the paddy field also. Okay. Oscillatoria have the capacity to fix nitrogen in paddy field. Cyanobacteria also lives in the symbiotic form. They make the blue-green algae forms symbiotic association with many plants and perform nitrogen fixation. Like nostoc found in the thallus of the hornwort. We'll talk about the hornwort while I will discuss about the kingdom Plantae. There I will discuss about the hornwort. So nostoc lives in association with the hornworts. If you know the Azolla, that is the pteridophyte, Anabaena lives in association with pteridophyte, that is Azolla, and Cycas, in the root of Cycas, Anabaena also establishes itself, and the root of the Cycas become coralloid. So the conclusion is these cyanobacteria may found in the free living as well as in the symbiotic relationship with the other plants. So if you want to discuss that what is the difference between the eubacteria and cyanobacteria have, we already know the difference between the archaebacteria and the eubacteria. Archaebacteria and eubacteria, there is many difference. Likewise, eubacteria and cyanobacteria have many difference. That's why we have kept into different subkingdom. The size of the eubacteria is small, however, it is comparatively larger. Eubacteria may have the flagella for the movement, but the cyanobacteria don't have. Eubacteria may be of autotrophic or heterotrophic, but cyanobacteria is always autotrophic. Reserve food material, just like we people keep our food in the form of the glycogen, the eubacteria also does the same. But the cyanobacteria keep in the form of the starch. Eubacteria may have the bacteriochlorophyll. It have the chlorophyll A just like the plant. It don't have any pigment, accessory pigment, but the cyanobacteria have the phycocyanin and phycoerythrin pigments. Now, the special category, I already told you that some bacteria also don't have the cell wall. So that bacteria is known as mycoplasma. Especially since it don't have the upper boundary as a cell wall. So obviously if cell wall is not there, then outer boundary will be the plasma membrane. So mycoplasma are genus of bacteria which belong to the bacterial class that is known as molecules, means absence of cell wall. And they are very simplest and the smallest free living prokaryotes. So it is very important in examination it may be asked which one is the simplest and the smallest free living prokaryotes. Your answer will be mycoplasma. As I already told you, mycoplasma, the cell wall is absent. Hence, the plasma membrane form the outer boundary of the cell. And due to absence of cell wall, the organism can change their shape also. So that's why they are called in nature they are pleomorphic, means they can change their shape. They are also called as mycoplasma-like organism or pleuropneumonia-like organism. So these are some name of the mycoplasma you can mug up. And first time they were discovered by the Nocard and Raux in pleural fluid. Pleural fluid means what? The bovine means cow. The cattle was suffering from the pneumonia. So scientists taken the pleural fluid which is present in the lungs and they found the mycoplasma. And it is the simplest and smallest known living organism. I already told you the size of the normal bacteria is 3 to 5 micron, and you can see the size of this mycoplasma is 0.1 to 0.5 micrometer, means much smaller than the normal bacteria, the eubacteria. Majority are parasite, although some are free living also. Let's understand how many they are causing the disease. So pleuropneumonia in domestic animals they cause, and in human they cause the infection of the urethra. So it is known as urethritis in human. In plant it causes witch's broom. Broom means jhadu, right, in Hindi. So this is the broom, witch's broom in the some plants. And in brinjal it causes little leaf disease. Okay. So one more special category that we have to discuss, that is known as actinomycetes. As it seems that they are fungi, but actually they are not fungi. They seems like fungi. They resemble fungi because of their elongated cell that branch into filament or hyphae. So if you see in the laboratory the colony of the actinomycetes, it will look like the fungi, but they are not true fungi. They are mainly found in the soil microorganism, and some are found in the marine also. They are gram-positive mycelial bacteria, means they have the mycelium just like the fungi. I already told you they look like fungi, but they are not fungi. They are actually bacteria, and they don't have any flagella. The actinomycetes produce many antibiotics and many medicines, chemotherapeutics, many compounds to kill the fungi, to kill the herbs, and compound is being isolated, that is immunosuppressant, means if you give that compound to any person, that immune system will be compromised, and that is useful because once the organ transplantation takes place, at that time doctor give the immunosuppressant so that patient can easily accept that organ. Okay, so since they are producing lot many compounds which is beneficial to us, that's why actinomycete is also known as natural pharmacist. One example of the actinomycete is streptomyces. So this is all about the kingdom Monera. Now we'll discuss about the kingdom Protista. So the general characteristics of the kingdom Protista, as already discussed, that all the prokaryotes were kept in the kingdom Monera, and all the eukaryotes, which is unicellular in nature, means they are made up of single cell, we kept under the Protista. So all the organisms included in the Protista are unicellular eukaryotes. But this Protistan, all the organisms being kept in the kingdom Protista, they are of the very mixed nature. Means some are having the plant-like character, some have the animal-like character, some have the fungi-like character, but actually not, they are not fully plant, animal or fungi. So what researchers have done, they have kept all the mixed type of characters of characters uh, organism in the kingdom Protista. So you can say that Protista are the connecting link between the bacteria and plant, animal and fungi, because the Protistan have all the characters similar to plant, some have the character similar to animal, some have the character similar to fungi. Okay, so all the members of the Protista are primarily aquatic. They found in the aquatic, means in the water. They are connecting link between kingdom Monera and the three kingdom like fungi, plant and animalia, as it is clear from the figure. So being prokaryotes, since they are eukaryotes, so obviously they should have the nucleus, well-defined nucleus, and all the membrane-bound organelle like mitochondria. If they have the plant-like characteristics, they may have the mitochondria as well. All right. Some have flagella or cilia for locomotion, and protist can reproduce with sexually or asexually by a process of involving cell fusion and zygote formation. So as I already told you that some Protista have the plant-like characteristics, although they are not plant. Some have the fungus-like character, some have the animal-like character. So while I was talking about the 7 obligate categories, I already told you that in the kingdom there is the, if there is the plant or fungi, we keep as a division, and in animal we keep as a phylum. So here this is the very interesting that under a single kingdom, that is the Protista, both are present. If they are plant-like, we kept in the division. Fungi, division. But if they are animal-like, we kept under the phylum. So this is the very unique example. In a single kingdom, we will find both the division as well as the phylum. So under the Protista, the plant which are the Protistan which are plant-like, we call them the photosynthetic protist, and there is total 3 divisions that we have to discuss, like Chrysophyta, Pyrophyta and Eglionophyta. The Protistan which have the fungus-like character, we have to read in the division Myxomycota, and the Protistan which have the animal-like characteristics, we have to study about the phylum Protozoa. So let's discuss one by one division. First we'll discuss about the division Chrysophyta. So Chrysophyta can be found in both freshwater and marine environment. All right. Chrysophyta is a diverse division. Lot many groups of Protistan are present. Here it is written groups of algae, but they are not true algae, they are plant-like. So just like diatom, golden algae and yellow green algae. So as name suggests, it is gold algae and yellow green algae. So you might be confused that these are algae, but actually they are not true algae. They are algae-like. So don't get confused. The golden algae, the yellow green algae and the blue green algae, which I discussed in the cyanobacteria, they are not algae actually. So each group has its own characteristics and ecological role to play, and most of them are photosynthetic in nature. We have to study about 2 group under the Chrysophyta, the diatoms and golden algae, that is known as desmid, in detail. So let's understand what is diatoms. Here you can find the different shapes, like some are circular, some are boat shape, some are triangular, right? Different shape of there. So these are the different shapes of the diatoms. The different diatoms having the different shape. They have the cell wall. So diatoms are maybe circular, maybe rectangular, maybe triangular, maybe elongated and boat shape. They have the plant-like character. Means their cell wall is made up of cellulose. But one thing is mixed here. The silica is the additional thing which make it shiny and very hard. So we call this cell wall as shell or frustule. Even after hundreds of years, once diatoms die, their cell wall remain as such. The cell wall doesn't get degraded. So that cell wall keep deposited in the surface of the bottom of the sea. And over the period of time, if diatomous cell wall is keep depositing, then it may found the rock-like structure, or that is known as diatomous earth. So diatoms are the chief producer in the ocean. It has the very fantastic role in the food chain in the ocean. And cell wall does not get destroyed, as I already told you, after their death. So at the bottom of sea, very huge rock of dead diatoms are formed, which are known as diatomite, diatomous earth, or kieselguhr. Cell wall of the diatoms are made up of two overlapping halves like a soap case. Just like in the soap case, one lid fit over the another. So the cell wall is known as frustule also. So it is made up of two things. The upper portion is known as hypotheca, downside is known as hypotheca. Upper side is epitheca, downside is hypotheca. It's just like soap case. You can find here is the very beautiful urn, just like we have to keep in the jewelry inside it. Okay, it is known as Melosira sulcata. Very how nature is fantastic. How much amazing things it have been created. How we can use the diatoms? Diatomous earth means if we keep in the powder form, and if the diatomous earth we can keep here and water is there, so water can be filtrated because small pore is there and water can be filtered through diatoms. Industrial abrasive or polishing. With the help of diatoms can be used as a polishing in the industry, polishing surfaces. Okay. Pest control. Since the edges of the diatomous earth, the cell wall is very sharp. So if naturally, if somewhere a diatomous earth have been spreaded and pest come, then pest will be hurted and gradually pest will die. So pest control can be done naturally by using the diatomous earth. Since the diatoms keep depositing on the sea bottom, so they ultimately can convert into fossil fuel, just like oil and natural gas. And diatomous earth is also used as cosmetics and personal care. Next is known as desmid. Desmid, they are also known as unicellular green algae. They are somewhat similar to algae Spirogyra. They occur in fresh water, and the unique thing with the desmid only grow in the fresh water where there is no pollution, means unpolluted water. So if in some water body desmid is found, means it is clear that is there is no pollution, that is the good water to consume. Now another one is division Pyrophyta. Under the division Pyrophyta, one class is known as Dinophysi. The organism being kept in the class Dinophysi are known as dinoflagellate. Their mostly members are unicellular, mostly marine and photosynthetic. There is two flagella, just like in, you can see one flagella just like here, and another flagella is wrapping around in the cell plate. They may appear, since this is the class, so many organisms are there. They may appear yellow, green, brown, blue or red, depending which type of pigment it have. Example of important dinoflagellates are Noctiluca, Gonilox and Gymnodium. Gonilox spreads on the surface of seawater, and through rapid multiplication it makes the seawater as red, so it is also known as red tide. Gonilox and Gymnodium both are toxic. They secrete one toxin. The name of toxin is known as saxitoxin. And these toxins cause paralysis in human being, and all the marine organisms are also get affected. They die due to the rapid multiplication of Gymnodium and Gonilox. Another dinoflagellate is Noctiluca. It is known as sea ghost and sea sparkle because in night it glow. So you can see a very beautiful picture here. Noctiluca is responsible for glowing of sea. Third is division Euglenophyta. The organism inside the division Euglenophyta is known as euglenoids. Majority are freshwater, found in stagnant water, means where water is not moving, right? Running water is not there, just like in the case of the river. So they may be found in the pond, they may be found in the lake. So previously euglenoids, means example of euglenoid is Euglena gracilis. Many species are there. Here I have kept only one example. So previously what happened, since the that Euglena have the photosynthetic ability, they have the chloroplast, they can photosynthesize. So scientists have kept in the plant kingdom. And once there is no sunlight, then it may turn into predator and it may eat the animal. So it is showing the heterotrophic character. So some scientists keep under the animal kingdom. So strange. It have both the characteristics plant as well as of animal. That's why they are known as connecting link between the plant and animal. Both are character, they are autotrophic as well as heterotrophic. So now since it have the mixed character, so now we are keeping into kingdom Protista. Well, they are unicellular. So if they are plant, then they should have the cell wall. Obviously, it should have the cell wall made up of cellulose, but interestingly, the cell wall is not made up of cellulose. Rather than cell wall, it have one protein-rich layer called pellicle. So one side, it is showing the character of the plant, like photosynthesis. Another side, it is showing the character of the animal, that is the heterotrophy. And again, it don't have the cell wall, means is showing the character of the animal. It has the protein-rich layer, which makes its body flexible. They have two flagella, one short and one long. And I already told you they are connecting link between the plant and animal because both characters it have. And interestingly, the pigment which it contains for the photosynthesis, they are identical to those as present in the higher plant. So Euglena is the best example of the euglenoids. Now fourth division, Myxomycota, means the organism present in the Myxomycota are known as slime mold. In NCERT, it is directly written slime mold. So slime mold is a very broad term. It applies many fungus-like protists. Means although they are looking like fungus, but they are not true fungus. Okay. So however, slime mold, which are the fungus-like protists, are now recognized as being different to fungi. They are called as false fungi. They look like fungi, but they are not fungi. Slime molds, let's understand the meaning of the slime. Slime means chipchipa, the sticky mold. So slime molds are saprophytic. They feed on twig of the plant or leaves of the plant. Mainly the colony they form during the rainy season where there is the plenty of water, humidity in the sea, humidity in the air. So under favorable condition, they spread the aggregate and up to several feet, you can see. And this is spreading of the slime mold is known as Plasmodium. Don't get confused with the Plasmodium vivax, which is the one protozoa, right? So Plasmodium means what? The colony of slime mold. So during unfavorable condition, what they do? The Plasmodium differentiate and it form fruiting body. It produces the spore which is spread in the air, and whenever favorable condition comes, they again grow and they form the Plasmodium. The spores are extremely resistant and survive for many years even under adverse conditions. The spores are dispersed by air current. Some of the example of the slime mold is Physarum and Dictyostelium. Now let's understand what is the similarity and dissimilarity that slime mold share with the fungi. Fungi is eukaryotic, no doubt. Slime mold is also eukaryotic. Slime mold, fungi is heterotroph, it is also heterotroph. They both have the sexual and asexual kind of the reproduction. They both have the sporangia where spore formation takes place. Then how slime mold differ than the fungi? The cell wall of fungi is made up of chitin, important character, but here the cell wall is made up of cellulose in the slime mold. This is the difference. Slime mold don't have mycelium, but true fungi should have. True fungi don't have plasmodium, but here it have the plasmodium. So this is the similarity and difference with slime mold and fungi. Now, some protista, as I already told you, have the character just like animal. So that we kept under the phylum Protozoa. So all protozoans are heterotroph and live as predator or parasite. They may live as a predator or they may act as a parasite. And they are believed to be primitive relative of animal. There are four major group of protozoans. So under phylum Protozoa, there are total 4 classes based on which type of locomotory structure they have. If they have flagella, we can keep them. If they have the pseudopodia, pseudo means false, podia means foot. If they have false foot, we keep under the rhizopoda or sarcodina. If they have the flagella as a locomotory structure, we keep in the mastigophora or flagellata. If they have the cilia, we keep under the ciliata. If they don't have any locomotory structure, we can keep in the sporozoa. So let's discuss each and every class one by one. So first we'll study about the sarcodina. Amoeba is the main organism in the class Sarcodina. They are known as amoeboid protozoan. They may be found in the fresh water, the seawater, the moist soil, and they move and capture their prey by putting out pseudopodia, false food. Say for example, here is the prey, and the amoeba is just like here only, because you know amoeba don't have any fixed structure. So gradually it will move its pseudopodia through cyclic streaming of the protoplasm. It will grab the prey and will eat it. So that is known as pseudopodia. Marine forms have silica shell also on their surface. Some of them, such as entamoeba, are parasite also. So this is all about the class Sarcodina. Here is the typical structure of the amoeba. Next class is zooflagellate, means they should have the flagella as a locomotory structure. They are either free living, found in the water or moist, or they are parasite also. They have the flagella for locomotion. Flagella may be one to many. Nutrition is holozoic in the free living. If they are free living, whole prey they can eat. And parasitic form cause disease such as sleeping sickness, caused by example is trypanosoma. Parasitic forms cause disease, many diseases like African sleeping sickness, one disease, it is caused by Trypanosoma gambians, and it is spread through the vector CC fly. This is the CC fly. Scientific name is Glossina palpalis. American sleeping sickness, it is caused by Trypanosoma cruzi. Leishmania donovani cause disease, Kala Azar, dum fever, visceral leishmania, and it is spread through sandfly. That is known as Philobottomus argentips. Giardia intestinalis, it is causing giardiasis, yeah, diarrhea, and also known as grand old man of intestine. The zooflagellata example is known as Trichonympha. It may also, lives in as a symbiont in the intestine of termite. Termite, you know, Dimak. It feed on cellulose. We cannot digest cellulose. But since the Trichonympha produce the cellulose or beta glucosidase, which can break the cellulose, that's why termite can digest the wood, means cellulose. Last one is that known as second last, that is known as class Ciliata. These organisms have the cilia for the movement. They are aquatic, actively moving organism because of the presence of thousands of cilia. Here you can see mouth-like structure, that is known as gullet or oral groove. With the help of that, it can eat the prey then how they can feed? Because they don't, we just like bee people, we have the hand, we can grab the uh, prey or food, then can eat. With the help of cilia, it moves in such a way, in the coordinated way, so that water comes inside the paramecium, and if prey is present there, that will enter inside the paramecium. Paramecium will digest the prey and remove the water, excess water. Example is paramecium. Now the last one is the known as Sporozoa, very notorious. This include diverse organisms that have an infectious spore-like stage in their life cycle. The best example or most notorious is Plasmodium. Plasmodium genus, but the colony of the slime mold was known as Plasmodium as well. Just like Plasmodium, let me write down, sorry, Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. Means these are different species of Plasmodium which cause malaria in the human, right? So it affects the human population. So this is all about the kingdom Protista. So Protista have all the three characters. Some are plant-like, some are fungus-like, some are animal-like. So all the mixed character organism we kept under the kingdom Protista. Now we'll study our third kingdom, that is known as kingdom fungi. So in the kingdom fungi, first let's study about its general characteristics. Students, the study of fungi is known as mycology. Fungi always prefer to grow in warm where temperature is more, but humidity also should be more. So the best season to grow for fungi is the rainy season, where temperature is more, humidity is more. Fungi may occur anywhere, in air, in water, in soil, and in the form of the parasite on animals and plants also. Most of the fungi are heterotrophic, and they absorb soluble organic matter from dead substrates and hence are called saprophytes. Means they are growing on the dead and decayed organism or plant. Some of the fungi, I also told you that they may live on the living plants and animals, and they act as a parasite. Moreover, they also can live as symbionts. Symbionts means what? As previously I also discussed that algae and fungi all together dwell and they form the lichen. This is the example of the lichen. Okay, algae do the photosynthesis and give the food to fungi, and in return fungi absorb the mineral and water and it gives to algae. So both are living symbiotically. In the higher plants roots, some fungi also may live as a symbiont, and that association is known as mycorrhiza. We'll discuss that later on. Here you can see, here is the plant. The fungi also live in the association. So what it gets from the plant gives the food to fungi, and in return fungi gives the mineral to the plant. So this association is known as mycorrhiza. As I already told you, the mode of nutrition, the saprophytic, they survive on the dead and decaying organism, and parasitic fungi, they live on another animal or living plant's body. Here you see the grasshopper, the fungi grows on it. The body of fungi is known as, the complete body of the fungi is known as mycelium. Mycelium is made up of many hypha. Hypha, you can see the unit from here to here. This is hypha, one hypha, the another hypha, the another hypha. So these are subunits of the mycelium is known as hypha. plural you can call as a hyphae. So mycelium is made up of hyphae. In many of the fungi, you will find that these two hyphae are separated with the one septa, one cross wall. So those fungi whose hyphae are separated by the septa, they are known as septate hyphae. And you will also see in many fungi there is no hyphae, although hyphae is there, but there is no separation of like septa. So those fungi are known as cenocytic hyphae. The important thing of the fungi is, as you know, the cell wall of bacteria was made up of peptidoglycan. The cell wall of plant was made up of cellulose, right? Likewise, the cell wall of fungi is form of glycogen. All right. So this is known as. So these are two characteristics, that the cell wall is made up of chitin, and the fungi stored their food, just like we people store our food in our muscles and in liver in the form of glycogen. In the same way, fungi stores its food in the form of the glycogen. So this is the similarity that fungi is sharing with the animal. As per reproduction is concerned, since the fungi is a diverse group of organism, so different fungi show the different mode of reproduction. So altogether, the reproduction methods can be categorized in two parts. One is the asexual reproduction, and another one is the sexual reproduction. So reproduction in fungi can occur through various mechanisms, including both it may be sexual or asexual. And since fungi are incredibly diverse organisms, and the reproductive strategies can vary depending on the specific species. If you talk about the asexual reproduction, so reproduction fungi basically produce some spore. Let's understand what is spore. Spore is a special structure which forms when unfavorable condition for the fungi makeup. Means whenever there is the unfavorable condition for the fungi, where fungi have the chances to be died, at that condition fungi produce a special kind of a spore. Means that is the asexual structure, single celled structure spore, and it is spread through air, water, or it may remain present in the soil, and whenever favorable condition come, it have the capacity to grow as a mycelium. Got it? So it's a totally asexual structure. So which are single structure capable of developing into new individual. There are several ways in which fungi can produce asexual spore. We'll discuss later on. So first method apart from the spore, although spore formation is the mode of the sexual reproduction, but some other methods of the reproduction is also there. One is known as fragmentation, as name says. Here you can see, here is the mycelium of one fungi, and if accidentally anyhow this mycelium break down, so this mycelium, this hyphae, will develop into new individual. So this is known as fragmentation. This kind of the mode of the reproduction was found in the fungi such as mold. Another method for the reproduction is known as budding. Budding, a small outgrowth called a bud. Here you can see this, the single celled eukaryotes, that it is known as yeast. It is also known as baker's yeast. So it start growing the outgrowth. Gradually, this outgrowth becomes large. It may form the chain of the outgrowth. This is known as the bud, and once it mature, it detach from the parent yeast and another yeast formation takes place as an independent organism. So this is method is found in the East. So this is the another method for the asexual reproduction. Now let's talk about the spore formation. Spore formation takes place in a special kind of the structure, and that is known as sporangia. So the organ you can say, or structure you can say in the fungi where spore formation takes place, that is known as sporangia. In some cases, the conidia formation takes place. Conidia is just like the spore. Okay. In the case of the penicillium, it forms the conidia. These conidia detach later on, behave like a spore, and after germination, they form the mycelium and finally the whole organism. Right? So sporangia are sac-like structure that contain spore, while conidia are free spore produced at the tip of the side of a specialized hyphae, that is filamentous structure. Means the difference between the sporangia, it's a closed structure where spore formation takes place. But in the conidia, it is open space where conidia formation takes place. It detached from the parent. These spores are then dispersed by various means like wind, water, animal, and can germinate under suitable condition to develop into a new fungi. Alright. Now another method is known as sexual reproduction. Sexual reproduction acts as there is no true kind of the sexual reproduction like male fungi or female fungi. The body of the fungi is always haploid. So if two mycelium come together, two different genotype or two different individual hyphae may come all together, they are specialized, then what is happening? Let's understand by the figure first. Say this is the mycelium. Another mycelium if comes, like I'm just drawing it, the another mycelium. What happens in some structures, two mycelium come closer, and these two I'm telling you, these two hyphae, these two hyphae may get fused. So once they get fused, what is happening? There is two nucleus, one nucleus is here, another nucleus is here. So in that case, once two hyphae get fused, their protoplasm or cytoplasm is going to be mixed, obviously. But so once the cytoplasm of both the cells mix, that is known as plasmogamy. Here cells from two different mycelia fuse to form a dikaryotic cell. Means cytoplasm get mixed, but their nucleus remain separated. So since in a single cell now there is two kind of the nucleus, that is why it is known as heterokaryotic stage or dikaryotic stage also. Now it depends on the type of the fungi. In some of the fungi what happen, once the plasmogamy takes place, meantime both the nucleus also get mixed and the cell become diploid. In some of the fungi, it remains separated for some time, maybe up to a week or more than that, and later on they get mixed. So ultimately they have to be mixed. In some fungi it quickly mixes, and in some fungi it delayed but later it mixed. So the fusion of the both the nucleus is known as karyogamy. Karyo means nucleus, gamy means together. So once both the nucleus fuse together, that is they form the zygote. So now zygote is 2n, diploid, and as you know all the fungi are haploid. So what happened? To make it haploid, meiosis takes place and spore forms with the help of the meiosis. So now this diploid zygote become haploid, and that is spore is spread in the air, soil, anywhere, and whenever the suitable condition come, it germinates. All right. You can see in the short, here is the one haploid mycelium, and you can say hyphae. Plasmogamy takes place, both the nucleus remain separated for some time, that is known as dikaryon. Later on, both the nucleus also get mixed. Now it is 2n, means synkaryon condition also. And now meiosis takes place, and from one cell, 4 haploid spore form. And these spore later on germinate and they make the mycelium. So very simple, how many events have takes place? First, the plasmogamy, means the mixing of the cytoplasm or protoplasm. Then heterokaryotic stage, means both the cells fuse, cytoplasm mix, but nucleus remain separated in a single cell. Karyogamy, later on, fusion of nucleus takes place, zygote formation happen, and later on, to make it haploid, meiosis takes place, and the chromosome number of cells become half. And this result in the formation of haploid cell called spore, and later on, once suitable condition come, the spore germinate and make the different mycelium. So this is the mode of the reproduction in the fungi. Basically, kingdom fungi, as per the NCERT, we have divided fungi in total 4 divisions. Although it is more, but here you have to read about the only 4 divisions. The Ascomycota, the Zygomycota, and Basidiomycota and Deuteromycota. So now, what is the basis for the classification of the fungi in the further division or in the classes? So as you know, that mycelium may have the septa, it may not. So the mycelium have septate or aseptate. What is the mode of spore formation? How spore formation takes place in different fungi? Whether the fruiting body is present or not. What does it mean by fruiting body? Fruiting body means what? The body of the fungi, in which form it may have the sporangia. You just check it out. Just like you might have seen about the mushroom you eat. Okay, so this is the mushroom. Once you open, you detach this part, you backside you see this like a structure. Here is spore formation takes. Sporangia is here. But once you start growing the fungi, it remain in the form of the mycelium. And once mycelium mature, then it forms the fruiting body. In fruiting body, sporangia is there, and then spore comes out over it. So all the fungi don't have the fruiting body. Obviously, those who don't have the fruiting body, they cannot have the sporangia, so they cannot produce the spore. All right. So bacteria, fungi may remain in two forms, either in the mycelial form or in the form of the fruiting body. So once you grow the mushroom at your home, first it will be in the mycelial form, and later on it will convert into fruiting body, and from fruiting body, sporangium will form, and from that sporangium spores will form and will come out, and again it will form the, suppose there is spore, again it will form the mycelium. So in all the fungi, doesn't guarantee that fruiting body is there. Fruiting body, maybe if fruiting body is there or not there, it makes the basis for the classification of the fungi. Okay. So the morphology of the mycelium, whether it is septate or aseptate, mode of spore formation, presence or absence of fruiting body, form the basis for the division of the kingdom into various classes. So there is totally basically 4 classes have been given in the NCERT. So let's discuss one by one. The phycomycetes. What does it mean, the phycomycetes? Once you read the phyco, it means sowing the seaweed. Means you can correlate with the algae. But you will be surprised that here I am talking about the fungi. Here I am putting the name as seaweed. So previously it was seen that they are sowing, they are just like the algae, they are floating in the water, So they're just like the symptoms of these members have just like the seaweed and mycetes, although they are looking similar, bit similar with the algae, but they are basically fungi. So all the algae looking fungi were kept in the class Phycomycetes, but later on it was found that this is not the true classification. So now the Phycomycetes forms especially spore, and that is known as oospore. That's why now class Phycomycetes is known as Oomycetes. So new name is Oomycetes, but in your NCERT Phycomycetes is there, so you can mug up as Phycomycetes or Oomycetes, means the fungi which contains oospore, means in which oospore formation takes place. So members are found in aquatic habitat or on decaying wood in moist and damp places, are obligate parasite and plants as well. A large group is there. The fungal filament, the mycelium of all the fungus included in this class are cenocytic, and aseptate. Obviously, if they are aseptate, means they are cenocytic. Means in one hyphae you will find many nucleus. Asexual reproduction takes place by spore in this case. Spores are of two types. If spores are motile, means they have the flagella to move, means for the locomotion, flagella is there. Those kind of spores are known as zoospore. And in some species, although spore formation takes place, but they are non-motile. So non-motile spores are known as aplanospores. And obviously, if spore formation is there, there should be some special structure, and that endogenous structure is known as sporangium, as already discussed. Two examples you can see here in this case. One is the Rhizopus, another is the Mucido. All right. Zygospore is formed by fusion of two gametes. So once the sexual reproduction takes place, so there is two type of gametes. If two gametes are of same size, try to understand. If gametes are of same size, means both are motile, look like similar, they are known as isogamous. But if the size of one is small and the size of another one is large, they are known as anisogamous. The size of both the spores. And if the one gamete is small, means motile, another one is large and non-motile, such condition is known as oogamous. So in the different fungi, different type of gametes are there, and based on that we call them isogamous. Iso means very simple, both the spores size are similar. For an isogamous, one is smaller and one other is larger, and in oogamous, one is motile, one is not motile. Okay. Some common examples you can say, that is known as mucor. Mucor, I have shown it. This is the mucor. During the corona season, you might have seen that people died due to fungus. So that mucor was responsible for that. That was known as a black mold also. Rhizopus, it is known as a bread mold. This is the bread mold. Once you keep bread in your kitchen and many 3 to 4 days you don't consume it, it rotten, and the Rhizopus infect over it. The albigo, this is acting as a parasite on fungi, on parasitic fungi and mustard. You can see here the plant of the mustard. Here the infection of albigo takes place, so definitely the crop yield is going to be decrease. Albigo candida. Now next talk about class Ascomycetes. Asco means what? Asco means sac. Sac means bag. Ascomycetes. So Ascomycetes produces spore, that is known as ascospore, within sac-like structure called SI, and that's why Ascomycetes member are known as sac fungi. They are mostly multicellular like penicillium, maybe unicellular as well, just like yeast. Baker's yeast, Ascomycetes contain both unicellular and multicellular. They may be saprophytic, they may be decomposer, they may be parasite also, or they act as a coprophilous. Coprophilous means what? The fungi which grow on the dung, cow dung, buffalo dung. Mycelium is branching, branch is there, and septate. They have the septa. Talk about sexual reproduction in case of the Ascomycetes. It is by conidia. Conidia is produced exogenously on conidiophore. On germination, produce new mycelium. Sexual reproduction. Sexual spores of Ascomycetes are known as ascospore, which are produced endogenously in the sac-like SI, and these SI are arranged in different type of fruiting body called ascocarp. Examples you can see: the Aspergillus, claviceps, and Neurospora. Neurospora crassa. Once I will teach you the genetics, there you will find that not many genetic related experiments are done by scientists are doing on the Neurospora crassa. So this is also known as Drosophila of the plant kingdom. All right. This is the example of this is the picture of Neurospora crassa. Penicillium is another example. This is known as blue or green mold. From Penicillium notatum, one very important antibiotic is formed, that is known as penicillin. So previously from the Penicillium notatum, penicillin was extracted, but nowadays more quantity of penicillin is obtained from Penicillium chrysogenum. Another antibiotic, antifungal compound we are getting from the Penicillium chrysophyllum. From the Penicillium camemberti and Penicillium roqueforti are used to produce the special type of cheese that is known as camembert cheese and Roquefort cheese respectively. So penicillium we also use for the cheese production. Aspergillus. This is the Aspergillus. They are known as black or brown mold or blacky smoky mold. One is Aspergillus proliferans. We get the antibiotic proliferin. Aspergillus fumigatus. They cause the disease in human and cattle, and the disease is known as aspergillosis. For example, aspergillosis of ear. If infection takes place in the ear, it causes the ear pain. If it reach into lungs, it shows the symptom just like the tuberculosis. Another is known as Aspergillus flavus. I'm sure sometime you are eating the groundnut, okay, groundnut or cashew sometime, and it gives lot many bitter taste. So you have to spit it out, okay. So that is actually the infection of Aspergillus flavus on the groundnut. It produces one toxin that is known as toxic substance that is known as aflatoxin, and that is potent carcinogen, means if it is used, it may cause cancer. So while you are eating the groundnut or cashew, you have to be very careful. There should not be Aspergillus flavus contamination, infection in that. So in case if it is bitter, you have to discard it. Another is known as Claviceps purpurea. Claviceps purpurea, it caused the ergot disease on the bajra. Here you see the bajra is known as pearl millet in English. So here you can see there is the infection of the Claviceps purpurea. One important drug is extracted from the Claviceps purpurea, that is known as ergotin. A narcotic drug, LSD. You might have heard about the LSD. Lysergic acid diethylamide is also obtained from it. And LSD is a hallucinogenic compound. Means if someone consumes this LSD, it may hallucinate, means the person will see the person actually who is died are not existing. So he will talk lonely with the person. LSD, hallucinogenic, means nothing is there, but the person who have taken the drug, he will assume with that person. Another is known as Markella. The species of the Markella are commonly called as morel, and it is an edible fungus. People used to have it in their food. It is a very delicate fungi, and the cost of 1 kg of Markela goes up to 50,000 rupees. Up to 50,000 rupees. Erysiphe, different type of species of Erysiphe cause powdery mildew disease on plant. You can see here the powdery mildew of wheat. It is caused by Erysiphe triticale. Erysiphe polygony, it causes powdery mildew of pea. And truffles, they are again the edible fungus. Yeast, as you know, it is used in the maximum in the bakery industry, for the brewing industry, to make the alcohol, means wine industry, and making the bread and synthesis of the vitamin B2 and all. So they are unicellular algae. Mycelium is absent in yeast because it is a single cell. Okay. So there is lot many applications of yeast. Next class is Basidiomycetes. They are commonly known form of Basidiomycetes are mushroom. Bracket fungi are puffball. They grow on soil and logs and tree stumps and in living plant bodies as parasite and they cause the rust and smuts. Mycelium is branched and the hyphae are septate. Asexual spore are not reported in the Basidiomycetes. Only vegetative reproduction takes place by the fragmentation, and the sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strain or genotype. The resultant structure is dikaryotic, which ultimately give rise to Basidium. So since Basidium is present, that's why it is known as Basidiomycetes. And karyogamy, meiosis takes place in the Basidium, producing 4 basidiospores, as I already told you. The basidiospores are exogenously produced on the Basidium. The basidia are arranged in fruiting body called a basidiocarp. So fruiting body is also there. Example you can see. Some common examples are agaricus, that mushroom, the well-known example which we eat. The astilago, that cause the smut, and Puccinia, that is known as rust fungus. Example you can see the rust fungi, the Puccinia. It may cause black rust or stem rust. This is known as black rust or stem rust. Puccinia recondita, brown rust or leaf rust. Puccinia striformis, yellow rust or stripe rust. So it is causing the many diseases in the plant. Ultimately, the crop production get drastically affected. Next is bracket or self fungi. These are epizalic fungi, means they like to grow on the wood. So on plant you can see they looks like bracket. Okay, just like bracket we are putting like this, right? So that's why they are known as bracket fungi. Example you can say Polyporus, puffball. Just like eggs, you can see the beautiful structure. Also, they are saprophytic fungi, means they are growing on the dead and decaying organism. Once you touch it, they will burst and the black color, they release the black color powder. Actually, there is the spore in this fruiting body. Example you see Lycopodium. That black powder is known as basidiospore. Sorry for my bad throat. Next is mushroom, which we eat. The button mushroom, which is known as Agaricus bisporus. World's most poisonous mushroom is known as Amanita muscaria, and from the Amanita muscaria, the compound which is very poisonous, that is known as amanitin. A very trace amount of this amanitin is enough to kill the people. And all the poisonous mushroom are known as toadstool. Some smut fungi now can see. The Astilago nuda triticale, it causes loose smut of wheat. It affects the wheat. Astilago madis, smut of maize. Astilago hordii, covered smut of barley. And Astilago cytomenia, whip smut of the sugarcane. So these are some diseases. Now next and last class is known as Deuteromycetes. In Deuteromycetes, mycelium is branched and septa is present. They are commonly known as imperfecti fungi. Why? Because the sexual reproduction is not reported in the class Deuteromycetes. Suppose today you found any scientist discovered some fungi and the sexual reproduction is not reported, so that scientist will keep that fungi in the class Deuteromycetes. And suppose after hours of research the sexual reproduction is also discovered, then that particular fungi will replace from the Deuteromycetes and it will be kept into a different class which they belongs to. All right. So the example is Trichoderma viridi, very important. It is acting as a biofungicide. Sometime what happened? The farmer face the problems of the fungus in their farm. So if they sow the Cajanus cajania, means you can say arahar in Hindi, or tuer dal, the pulses. So pulses wilt once they get mature, and the farmer don't get the production. So in that case, if trichoderma, if they spread the Trichoderma viridi in their farm, so what happened? Trichoderma viridi makes its mycelium, and it hinders, it stops the growth of other fungus in the farm, which is harmful to the crop. So this is naturally, and Trichoderma viridi does not harm the crop. So that's why it is known as biofungicide. Many other examples are also there. Many are parasite and plant and causes disease. Some members are saprophytic or parasitic. Large number of members of this class is decomposer and help in mineral cycling. Example is Alternaria, Coleotrichum and Trichoderma. This is the Coleotrichum, this is the Alternaria, and Trichoderma viridi I have shown already. So these are the examples which have been given in the NCERT. The Phycomycetes, also known as lower fungi. 3 examples was given: Rhizopus, Mucor and Albigo. Sometime in the NEET examination it is asked that what is the class of Rhizopus or Mucor or Albigo. You should remember the Phycomycetes. For the Ascomycetes, there is total 4 examples being given in the NCERT. The yeast, Aspergillus, Penicillium, Neurospora. Basidiomycetes is known as club fungi. The Agaricus means your mushroom, Polyporus, Puccinia, Osteologa and Lycoperdon. Deuteromycetes, Alternaria, Coleotrichum, Colectotrichum and Trichoderma. So these are different examples being given in the NCERT. I hope your fungi, the concept of the fungi is being clear now. As you know that maybe it is 5 kingdom classification or 6 kingdom classification. Scientists have not placed the virus or prions or lichens in any of the kingdom. So still these organisms don't have, you cannot say they are organism, but these entities don't have any place, any kingdom to put it. So that's why away from the 5 kingdom classification, I'm going to teach you the virus, prions and lichens separately. All right. So let's understand the virus. What does it mean by the virus? Now after the corona pandemic, everybody knows about the virus. Let's discuss in bit detail about the virus, lichen and prions. So virus is a Latin word which means poisonous fluid or venom. One scientist, Dmitry Ivanovsky, he recognized that certain microbes are there which are the causal organism of the mosaic disease of tobacco. Here you can see the healthy leaf of the tobacco, and here you can see the leaf affected with the mosaic disease. All right. So he found that bacteria is basically not responsible for this infection. So these were found, and the bacteria was found that spores are smaller. The viruses are much smaller than the bacteria, and even they can pass through the bacteria-proof filters. One scientist was there, M.W. Buzernik. First time he demonstrated that the extract of what he did actually, let me show you some experiment performed by M.W. Bejernik. Suppose you take three leaf. Okay, so this is the leaf number one. This is the leaf one, and it is infected with the tobacco mosaic virus. Okay, it is infected. So what Bejernik did, and these two leaves are healthy leaves. They don't have any kind of the infection of the virus. So what he did, first time, what he did, M.W. Bejernik, he extracted the infected, he extracted the soup. Soup means you just mash up these leaves and whatever extract comes. So that extract he poured on the healthy leaf. After some time, he observed that this leaf is also infected because the extract of this infected leaf was poured on the healthy leaves. Then what he did? Again he taken the extract and he kept in one bacteria proof filter paper. Means bacteria cannot cross this filter paper. Then whatever filtrate comes, that he poured on the another healthy leaves. Surprisingly, this leaf also got infected. So one thing was quite sure, that here bacterial infection is not there. So something is there which is smaller than the bacteria, which can cross the bacteria proof filter, and then that organism, the pathogen, was being known as virus. So this is much smaller than the bacteria. One scientist, W.M. Stanley, in 1935 he showed that viruses can be crystallized, and crystal consists largely of proteins. Means outside the host they are behaving just like a crystal. The non-living means if outside it is being thrown, virus don't show any kind of the living organisms symptoms. So virus, once the virus is not in the living system, virus only acts once it infects the living cell. So virus outside the host cell is known as virion. So there is no difference in the virion and virus. If virus is outside of the cell, it is known as virion, and once it is inside the living cell, it is known as virus. All right. So what are the different characteristics? These are non-cellular organisms. There is no concept of the cell here. The nucleus is there, nucleolus is there, mitochondria is there, nothing. They are obligate intracellular parasite, means they will show only the living character once they infect the cell, they are inside the cell. But one thing they have, RNA or DNA. Just like we people, we have the DNA as a genome. Likewise, virus may also have the RNA as a genome as well as DNA as a genome. They are inert outside. As I already told you that outside the cell, they are showing all the characters of the non-living, and they are very specific for the host cell. Means if some, just like take the example of the coronavirus, we are being affected by the coronavirus only can affect us. It does not affect the dog. It does not affect the cattle, right? So they are specific. So the coronavirus will affect only us, not to other organisms. Likewise, all the viruses are very, very specific to their host. If plant virus is there, it is not going to affect the animal or to us. They contain nucleic acid. So obviously they are capable of protein synthesis by the help of the ribosome of the host cell. Actually, it don't have the ribosome and all the machinery for the protein synthesis. It don't have anything. Virus have only and only RNA or DNA. And what happen if any living cell, it infect any living cell, then it hijack the mechanism of the living cell, our cells. It hijack, and whatever protein it want to synthesize, it will synthesize by our own machinery. Means the living cell synthesize the protein of virus. All right. So it just like take the example of the one example, typical example. Suppose any terrorist is there. A terrorist go to any home and they make the hostage to the family members and they demand cook food for us. So obviously, if they are on the gunpoint, they have to cook it. So the same way virus is doing. Once virus infect the living cell, it take the control of the living cell, and whatever the need of the virus is to multiplicate the virus, all the proteins, it uh, it you know it forms with the help of the host cell. Okay, so it replicate only inside the living cell of an organism. Morphology, if you see, the size of the virus is much smaller, just like the bacteria, it was 3 to 5 micron, but the size of the bacteria, the TMV, tobacco mosaic virus, one of the example, that is 300 millimicron, means thousands time or more than that, it is lesser than the normal bacteria. If you see the shape, in the virus presents are in different shape, like spherical, you see the influenza virus, the HIV, human immunodeficiency virus, which cause the AIDS in the people, in the human. It may act as a rod shape also. It is present in the rod shape. Here is a tobacco mosaic virus. It is in the rod shape. It may form as a tadpole-like structure. See the tadpole. Example is bacteriophage. So what is the chemical composition of a virus? Virus may have either DNA or RNA. So there is no fixed rule. That sometimes it is written that plant viruses have always single stranded RNA is present in the plant viruses. And in animal viruses, double stranded DNA is there. But exception is always there. Like cauliflower mosaic virus, it have the double stranded DNA. Some animal virus, RNA is also present, which is single or double stranded. So there is no fixed rule. Just like we people, we have the genome as DNA is a double helical, right? And RNA is a single stranded. But some virus have double stranded DNA, some virus have single stranded DNA as a genome also, some virus have single stranded RNA as well as double stranded RNA also. So lot many varieties are present in the viruses. Here you see, here the genome is DNA, here what is? Here the genome is RNA in the HIV retrovirus, and the adenovirus, the genome is DNA. Okay. Examples, you can mug up some examples like double stranded DNA virus, hepatitis B virus, they cause the hepatitis. Single stranded DNA virus, tomato pseudocurly top virus, double stranded RNA virus, rotavirus, which cause the diarrhea in the infants, in small babies. Single stranded RNA virus containing viruses are tobacco mosaic virus, HIV, polio virus, coronavirus. Then out inside the genome, means inside the core of the virus, there is the viral genome. Maybe it is single stranded DNA, double stranded DNA, single stranded RNA, or double stranded RNA, depends on the virus. Outside of it, one capsid is, one protein coat is present. Here you can see protein coat, and that protein coat is known as capsid, and it is made up of simple units, small subunits, and that small subunits is known as capsomere. So capsomere, many capsomere all together makes the capsid. Okay, in some bacteria and the viral genome and capsid are all together combinedly, we call it the nucleocapsid. All right, some virus, there is additional covering, and that additional covering is known as envelope, which is made up of protein. So those viruses which have envelope, they are known as enveloped virus. And some viruses also have another spike proteins. This is known as spike proteins. You might have seen the structure of the coronavirus. So it have the crown-like structure. That's why it is known as corona. So spike proteins, they are known as corona. Crown, they're similar to crown in the case of corona. So that's why the name was kept as a coronavirus. So viruses are of maybe 2 to 3 types based on its host. So some viruses affect the plant virus, affect the plant. So they are known as plant virus. We don't have to bother with the plant virus. Plant virus is not going to affect us. Example, tobacco mosaic virus, obviously it affects the tobacco. Banana bunchy top virus, it will affect the banana's growth. And tomato leaf curl, as name suggests, the leaf of the tomatoes will curl, and ultimately the tomato plant will get affected and it will give the less yield of the tomatoes. The animal viruses which affect us like polio, smallpox, influenza, rhinovirus. Rhinovirus is basically responsible for common cold in us. Seasonally, we get affected with a common cold. So rhinovirus is responsible. And the good part is some virus infect plants, some virus infect animal, and some virus infect bacteria also, means they kill the bacteria. They are known as phages. Like in the Ganga water, the bacteriophages are there. That's why all the bacteria being eaten by bacteriophages. So they are good for us. Some of the pictures, beautiful pictures you can see. This is polio virus, this is influenza virus, this is rhinovirus, this is smallpox virus. Bacteriophage virus you can see. The virus which infect the bacteria are called bacteriophage, and they have the double stranded DNA. And but in the another phage, 174 bacteriophage, here it have the single stranded DNA. Now another term that is known as viroid. What is viroid? As I already told you that the bacteria may have the, virus may have its nucleic acid, it may be DNA, it may be RNA, right? And out of it, one protein coat is there, that is known as what? Capsid. And outside of it, envelope is there. Sometimes also happen, some virus also don't have the capsid, only RNA is there. Necked RNA. Outside of it, the envelope may be there. So if capsid is not present in the virus, that is known as viroid. The only difference between the virus and viroid is virus have the capsid, means it is made of the capsomere, but viroid don't have capsid. They lack capsid. It was discovered by Theo Diner. Example is they cause potato spindle tuber disease, that is known as PST viroid. Next, prions. What are the prions? Prions, if you see in the books, there is the beautiful structure of the protein. And every protein have their specific 3D structure. If somehow this 3D structure get unfolded or abnormally folded, properly it doesn't fold, it may cause infections. So abnormally folded protein may cause the infection to others. That is known as prions. So prions are nothing, they are proteins only. And they were similar size to viruses. How is what discovered? Some tribes was there. They saw the cannibalism. Cannibalism means human eats human. Right? In tribes, sometimes it is reported that some people eat the people. So they started the dead humans, or they kill the human and they eat. So one very abnormal disease, they infected with the very abnormal disease, and later on it was being found that the abnormally folded protein was eaten by these people. They got infected, and that is known as Creutzfeldt-Jakob disease. This is known as laughing disease. People behave like a mad and they become die. The mad cow disease. In the cow also it was reported. So prions are abnormally folded protein, and it basically comes with the cannibalism. Next is lichen. As I already told you, lichen is made up of the association of the algae and the fungi. The part of the algae is known as phycobiont or photobiont, and the fungal component is known as mycobiont. As I already told you many times, that fungi gives the mineral and water to algae, and algae perform the photosynthesis and it make food. And both live in the symbiotic relationship. So this is all about the chapter number 2, that was biological classification. Hope that chapter number 2 is clear to you. If you have any doubts, you can ask me the question as well. And if you're watching me on the YouTube, please do subscribe, like my channel, and please share with others so that the knowledge is spread to all. Thank you so much. If you want to join the live classes, you can download our application Sri Vishwakarma Academy, which is available on the Google Play Store, and you can take live lectures as well, the notes, the test, and everything. Thank you so much.